Hello, my name is Carmen Resnick, and today I'm here to talk about my work studying the sequence function landscape for antibiotic resistance in the dihydrofolate reductase family. Now, this is a really lengthy title, but what this really means is that I'm looking at how mutations to a single gene influence its ability to resist the antibiotic. Now, why am I looking at antibiotic resistance? Antibiotic resistance, as the name implies, is what occurs when the bacteria is able to actually fight back and resist the drugs that we treat them with. When this happens, this resistant population is able to actually dominate and take over the susceptible species, leading into an era where we're actually unable to treat bacterial infection with traditional medicine. For this reason, the Center for Disease Control has called this a global public health threat. This affects everyone. The Prime Minister of the UK, David Cameron, says that if we fail to act, we're looking at an unthinkable scenario where antibiotics no longer work and we're cast back into the dark ages of medicine. In 2019 alone, we saw nearly 5 million deaths associated with antibiotic resistance worldwide. And most importantly, it affects everyone of any age in any country. For this reason, it's been a primary focus for a lot of output articles in the recent decade, including one that I saw quite recently, just at the end of April, in Seattle, 4 dead and 31 infected. Now, this makes it a really urgent issue to face head on, and some current solutions include limiting the use of antibiotics in the first place, taking them out of livestock and agriculture, limiting their use to only the really necessary infections and medicine, and making sure or discovering new ways to contain these outbreaks of resistant bacteria when they do occur. What I'm looking at, however, is taking a step deeper, and I'm looking at actually studying the genetic sequence that's allowing these species to develop resistance in the first place. And we're doing this by using what's called a broad mutational scan, which I'll get into later. But what that means is we're looking at how nearly any possible mutation, even ones that don't exist in nature yet, can confer resistance. And this allows us to inform future drug design as well. Now, I'm studying specifically this enzyme dihydrofolate reductase, or DHFR. As the name implies, DHFR reduces dihydrofolate to tetrahydrofolate. When this happens, THF is then a primary player in the DNA synthesis pathway, making this a primary target or necessary for some cells that are rapidly dividing, like bacterial cells and cancer cells. This makes DHFR a target for chemotherapeutics and antibiotics, like methotrexate, which targets human cells, and trimethoprim, which targets bacterial cells. If we take out dihydrofolate reductase, we take out tetrahydrofolate, we take out DNA synthesis, and the cells can no longer continue to divide. We've done this study using a really primary facet of this lab, which is DropSynth. DropSynth allows us to actually synthesize huge libraries of DNA for a very low cost that are very diverse. And this is what's allowed us to actually conduct this study, looking at all of these mutations to the single gene. The library that we're working with has about 1,500 unique homologs, and within each of these homologs, there's an average of about 187 mutants. This makes this study comprised in total of over 20,000 unique variations to this DHFR sequence. This study is the largest study done in this platform to a single metabolic enzyme. What's unique about this study and what's unique about DropSynth is this diversity of the library. And this is really demonstrated on this figure on the left, which shows how all of the sequences in our library relate to wild-type E. coli, the focus of the study. Shown in red at the top here is wild-type E. coli. And you can see that the majority of this library actually shares less than 50% of amino acid homology to this E. coli sequence, shown by this purple ring around it. Now, I'm going to briefly run through how I'm actually conducting this study. We've started with this DHFR library generated through DropSynth. This library has barcodes, short 20 base pair fragments at the end of the sequence. And we've then taken this library and inserted it into a plasmid that allows us to express the library in E. coli. We've used first a generic strain, and then again transferred this library of DNA into what's called a knockout strain. And the knockout strain has both dihydrofolate and its partner thymidylase synthase knocked out of the strain, meaning that when we put this DNA into the cell, we are forcing the cell to rely on our library in order to survive. So we've exposed these cells to non-supplemented media, meaning it's using DHFR to survive and create these metabolites that it needs, and also exposing it to trimethoprim in a gradient so we get a full range of how well these cells are actually surviving. And finally, we analyze the data in this broad mutational scan. Now, how are we actually processing our data? As I mentioned, we use these 20 base pair barcodes. We can take them out of the study so we can see, with using only a very short fragment, how many times every single one of these unique species are present throughout the study, both from start to finish. We recover how many times they're present at the beginning and end, and then assign a fitness score. And this fitness score is calculated by looking at how many barcodes are present both before and after selection. So that's before and after complementation in E. coli, and then also after antibiotic selection. A positive fitness score means that the cell is increasing in growth and surviving very well, deemed enrichment, and then a negative fitness score is depletion. So what this looks like for the grand sum of our study is plotted out here. First is this complementation. Without any supplementation and without any trimethoprim, how much of the cell can actually survive, how many of these cells can survive using all these unique DHFR variants. We see that for the most part, it's pretty stable, shown by that 0 log change. But as we move down, we start to see this graph shift to the left. And this is an increased trimethoprim, meaning that we're seeing more depletion of the species, which is what we expect, is we'd see more species start to lose survival abilities in increasing inhibition. However, what we're really interested in is looking at these species on the right-hand side with the positive fitness change, and these are our antibiotic resistant species. Now I'm going to talk a little bit about what the bread and butter of this study is, which is the mutational scan. A deep mutational scan, I'll explain using a little bit of terminology. Take a sentence, a wild cat ran fast. The sentence makes sense. It has everything that it needs to be a complete sentence. And we can plot it out onto this map that shows us how all of these different variations are, or how each of these positions for letters influence its ability to be read as a complete sentence. Say we take this sentence and we exchange the F for a P. A P in the sentence makes the sentence a wild cat ran past. And this is now assigned a positive fitness score. This means that in the context of this sentence, a P in that position makes more sense to the sentence. But if we exchange the C for an H, we get a wild hat ran fast. It still is a complete sentence, but it makes a lot less sense. So it gets a negative fitness score. And then finally, what if we have a letter completely knocked out of the sentence? A wild cat and fast. This now doesn't make any sense at all. There's no verb in the sentence. And so this gets a very low fitness score, telling us that the sentence no longer makes sense in the context that it's placed in. And so we've conducted this exact method, but with the amino acid sequence for these genes. And furthermore, is we've expanded this into a third dimension. This is where it gets a little complex, but what this means is that we're looking at how all of these homologs, which have very different sequences, are able to collapse on top of each other and compare to one another in what's called a broad mutational scan. So I've created a broad mutational scan for this library that I'm focusing on. Here we see the complementation assay. How well do these sequences relate to E. coli? And you see in the green boxes is the wild type E. coli sequence. For the most part, things are surviving really well, as shown in yellow. Some things aren't present at all, shown in gray. And then we see our depletion and enrichment with red and blue boxes, as denoted in the figure in the scale down here. Now, we've done this again as well, only with a high trimethoprim inhibition concentration. And so what this shows us is a lot of these species are not able to survive in E. coli against trimethoprim, as expected. However, there are a few that are able to have a fitness score, meaning they are actually present in the study, either at a lower or higher concentration, dependent on that actual fitness value. So the points that we're really interested in on this map are these red and orange and yellow spots that tell us that there's complete survival of this mutant when that amino acid is present. And so this chart doesn't tell us a lot until we actually map it out onto the DHFR molecule. So here you see this complementation map, and we can see that a lot of mutations are tolerated pretty well, shown in white. And we can look at that at the 3D surface as well. Here, the pink molecule in there is our DHF. Now, I've done the same thing, however, for trimethoprim inhibition. And you see that a lot of these sequences are not very well tolerated, shown by a large amount of blue, especially on these surface areas. But there are a couple of pockets that we see in red that infer this survival and increased survival with some mutational change. And these are what's interesting to us. Here, pink in the pink molecule is actually that trimethoprim. Now, how can we actually read this map? If I take a single point, so this blue on our complementation assay, meaning it kind of survives but not very well, changes to red under trimethoprim inhibition, and this tells us that there's actually an increase in survival against trimethoprim, and that's of interest to us. So we can narrow in on that point, and through some methods, I'm able to find that that position corresponds to T46. I know that by tracing to the wild type position on this high inhibitory concentration map. And then I can look now vertically from that position, and I see that there's a couple of red and orange spots. So this particular species might correspond to the mutant T46N, where an N has replaced that T in that one position. And this might be a resistant species. And so we can do this now for a lot of the different variations on this map and learn from it. And this actually allows us to apply this information into creating new drugs that might mitigate that single location to improve resistance and actually resist resistance. Thank you so much. I appreciate you taking the time to hear me speak. I'd also like to thank the Plesha Lab and the cohort of the Knight Campus undergraduate scholars, Dr. Kaylin Plesha, Samuel Hinton, and Professor Brian McWhorter of the Clark Honors College, and of course, to all my friends and family for support.